Good morning, everybody. You've made it to the second to the last talk, so you're almost done. It's good. We're getting there. So how do I move this thing?
The green button.
Ah, the green button. Okay.
Yep.
So, as you've seen this slide at each of the talks that are kind of come along, we're now to the second to the last talk, and for some reason they gave me rare meat. And so I'm actually going to utilize that theme throughout the talk. So if you're vegetarian, I apologize. I don't have any conflicts of interest that I need to disclose for this. So we've seen this slide a couple of times. This is Jason Stein's paper from 2012, where a significant number of individuals, where we were able to generate hippocampal volumes from a significant number of individuals, and then we perused the genome over 500,000 or so locations, and we came out with some hits that gave us a locus, an area in the genome that seems to be influencing hippocampal volume, right? And then we've also seen this other slide a couple of times today, which is Derek Hybar's paper, which was then in 2015, that was kind of doing the same thing, now with a larger sample and looking at lots of different brain regions. But there's some important things that we need to think about with this. One of them is that what these types of studies, and basically all GWAS type studies, are doing is they're giving you a locus. You are found a point in the genome that is indicating that a gene may be harbored somewhere nearby that's influencing the trait that you're interested in. It's not a gene. And Derek will spend a lot of time in his talk trying to explain to you how you can go from this locus to a gene. But there are a couple of points that we have to think about. And one of them is really just kind of thinking globally, what's the point of genetics in general, right? So there are different types of points, but classically, what you want to do is try to find a gene or a set of genes influencing a complex trait. And you do that first by finding a quantitative trait locus. And that's what you've done with the GWAS study. You found a locus somewhere in the brain, somewhere in the genome, and you've said, okay, here we have a locus. The next step from that is to look for identification, is to go from localization within the genome to identification, so trying to find the gene that actually seems to be involved. And the third step there is then to actually go in and look at the exact nucleotides and try to figure out how that's working within this effect. So this is sort of the classic, how do we do genetics in a classical form, right? Okay, so the point is that the work that we've been doing thus far has really been primarily accumulating QTLs. Right? So there's an estimate that we have about 2,000 loci that have been identified through different GWAS studies to date. Actually, this was about 3 years ago when this estimate came out. So we have a lot of loci, but very few of those loci have actually been turned into true gene identifications. And there's some reasons for this. One of them is that you're using as I said before, you've identified a locus. And if you're doing this with association, you've identified a locus that's about 500 kilobases, right? So in a 500 kilobase region, you could have scores of genes, right? If you're doing it in the more classical linkage sense, you could have a region that was between 10 to 15 megabases, so much, much bigger, where you could have hundreds to thousands of genes. So the issue is we've identified a locus, and now we need to go from that locus to the gene. The problem is that identifying the underlying genes requires certain assumptions or certain types of additional work, or more classically, doing sequencing and looking within that region. And I'll give you an example of this. This is a classic example from the Schizophrenia Genetics work group for the Psychiatric Genetics Consortium. So they took 40,000 or 36,000 individuals with schizophrenia, and they compared them to 100,000 or so controls at millions of points on the genome, and they came up with 108 loci that dissociate individuals with schizophrenia from controls, right? Now, none of these loci is giving you a specific gene. They are giving you a locus. But you can see that you have this one mega hit right here, right? This really big guy. Well, the point is, we've known that for a long time. We've known that locus for a long time, but we haven't been able to figure out what the gene or genes in that locus might be. And there's a reason for it. One of them is that locus is in the major histone compatibility complex, which is an area of the genome where it's very, very complex. There's a lot of kind of overlapping genes. There's a lot of linkage disequilibrium. It gets fairly difficult. But then this paper last year came out from Steve McCarroll's group where they went in to carefully look at that locus in large numbers of individuals, and they were comparing individuals to their parents and trying to work out particular haplotypes, and they found a haplotype on one gene, the complement component 4 gene, or the C4 gene, and they showed that the C4 alleles that were associated with schizophrenia were associated with greater expression of C4a, which is one of the splices or one of the kind of variants of this gene. And the nice thing about it was that they could then put that into a mouse model, and they showed that alteration of that C4 mediates synaptic elimination during postnatal development. So it influences pruning, and there's been a pruning hypothesis of schizophrenia for decades. So everybody looked at this and said, this is wonderful. Right? We may have a gene that is part of that locus. The thing, though, that you have to realize is that it's not explaining all the variants in that locus. It's just explaining a portion of the variants in that particular locus. But this is the kind of work that rarely happens in psychiatry, which is that you go from the locus to try to actually figure out what the gene might be. And it's also rare or never in imaging genetics, where you're trying to push to that next level. And there are a number of reasons for that. And part of the reasons are that it tends not to be that we find effects that are as large as you might need in order to be able to do that next search. So here's my first random red meat picture. So why study rare variation? So there are a number of reasons to study rare variation, partially because I like it, but also because you have an argument with possible effect sizes. Right? So what do I mean by this? Well, if you have a rare monogenic disorder, you can find one locus that gives you all of the heritability of that disease, right? You have one gene, one locus, it's explaining the entire heritability, and you can model that in standard deviation units, so displacement units between the people who are monozygotic versus, who have the allele who don't have the allele. Okay. So that's the classical sense, right? On the other end, you have GWAS-derived or common variants that we're looking for. And there, you tend to find for any specific variant, you're explaining very little of the total heritability of the trait, right? So often less than 0.01%. Okay. And the mean displacement associated with them, or in this case, effect sizes, much as JB was showing us before, is often relatively small. It's about 0.15, right? There's a lot of variation. It depends on how big the effect is, but it's relatively small. So the problem is, if you have an effect that's that small, it's very hard to go into the lab and do the functional biology and the functional genomics to convince yourself that that's the hit you want to follow, right? In contrast, there's a group of possible effects in rare partially penetrant variants that can give you a reasonable amount of heritability in those individuals that have it, so in a given particular pedigree. And the effect size you would be expecting is between 0.5 and about 3.5 standard deviations, right? So this isn't to say that all rare variants are doing this. This is to say that there is a population of rare variants that you could find in the genome that would be allowing you to look for something like this. And if that's the case, it's much easier to convince yourself you now want to do functional biology to try to work out what exactly is the gene that's influencing us here. Okay, so we're going to talk about rare variation then, but we're going to start not by talking about rare sequence variation, but we're going to start by talking about rare structural variation. And in this case, we're really thinking about copy number variants. So a copy number variation, or CNV, is a structural variation involved in an unbalanced rearrangement of DNA segments, which can alter the diploid status of the DNA. Okay, so you can have microinsertions or microdeletions or duplications, and the number of duplications or the size of the deletion can vary by an individual, right? So there are a lot of issues here. We've really only been studying CNVs very carefully for about the last 12 years or so, and so the definitions change a little bit. What they usually change in is how big the CNV has to be before we can start to look for it, right? In older days, when we had sparser arrays, CNVs had to be larger for us to track them down. Here, based on a recent paper from Hastings, a recent paper, they were kind of arguing that you can think about a CNV as being slightly larger than 50 base pair, right? And if it's less than that, they want to call it an indel. It's kind of an arbitrary threshold at which you're putting it at. And when we start doing this in the sequence data, there's probably going to rearrange how we think about it. Okay, but there are a couple of things that are important to realize. A CNV, so in this microdeletion or microinsertion, has a number of different possible scenarios. It could have no effect on your phenotype. It could be adaptive, so it could be a positive effect on your phenotype, or it could be negative. It could cause a disease, right? And there are different data for each of these things. And so I wanted to kind of give us a little bit about that. There's also the fact that there's been a lot of work done to show how CNVs are influenced throughout the genome, because they're not randomly distributed. There are kind of hotspots or areas in the genome that seem to be more likely to have CNVs present. And this recent paper went in and looked at, I think, 300 or so individuals that they had sequence data on, and they said that somewhere between 5 and 9.5% of the genome it contributes to CNVs. Okay, so we have a smaller subset. They tend to be relatively rare, which is why I'm talking about them in a rare variation session. So what do we know about CNVs and their relationship to brain-related diseases? So the first was a big paper by Jonathan Sabat and colleagues in 2007, where they showed that CNVs were associated with increased liability for autism. Subsequent work has suggested that about 5% of the cases with autism have a relatively large chromosomal rearrangement or a CNV. They're both de novo, meaning they're CNVs that only exist in the affected individual, not in their parents. And recurrent CNVs are associated with risk for autism. And here's a list of locations where we tend to find those CNVs. I'll point out two of those locations. One of them is the 15q11 location. The other one is the 16p11 location, because those will both come up again. In schizophrenia, there was just a paper that came out a couple of weeks ago, actually, that showed that went in from the Psychiatric Genetics Consortium and looked at 20,000 or so schizophrenics and 20,000 or so controls and modeled all the CNVs that they could find. And they found 8 that were genome-wide significant, given the fact, because they occurred enough times in that sample in controls, but not in patients. And so what you see here is you get some of those same deletions that were associated with autism, but again, you're kind of working it through. And here, particularly, you get the biggest effect is at 22q11, which we'll come back to in a moment or two. Okay, so does this affect brain and cognition? Well, the answer is yes. You have dose-dependent brain and cognitive alterations in that 15q11 CNV that's associated with schizophrenia, or at least it's associated with schizophrenia if you happen to be in Iceland. And so what you see is that individuals who have those deletions in that CNV are also show cognitive impairments and alterations in neuroanatomy that are similar to what you're seeing in schizophrenia, even though the individuals they were studying here did not have the illness itself, right? So that means that these structural variants are influencing brain in the kind of the things that we're interested in. And then there was an incredibly eloquent study by the group at Harvard where they were looking at both deletions and duplications. And so the question was, if you have, if you look at that, one of those loci that's been associated with both schizophrenia and autism, the 16p11 locus, and you say, okay, well, if there are deletions and duplications in that region, do they have any influence on neuroanatomy? So what they did is they found 25 individuals who had a deletion and 27 individuals who had a duplication, and they had matched controls, and what they showed was if you have a deletion, you have larger intracranial volume, larger brain volume, more gray matter, more white matter, and more surface area, but not more thickness. In converse, if you have a duplication, you have smaller of all of those things. So what you're looking at is an opposing, a relationship where you have deletion, control, duplication, right, in the size of brain. So again, it's telling you that rare genetic events are influencing brain in much the way that we would anticipate. Here's a paper that just came out from Carrie Bearden's lab at UCLA, where they were looking at that 22q11 data, and these are very similar to the data that Derek presented earlier, although he was presenting from the Enigma Consortium. This is just from their work at UCLA, and you're seeing a similar sort of pattern. That is, the 22q deletion individuals, in this case, are showing lower intracranial volume, lower gray matter, lower total white matter, and the duplications are showing higher, right? So, okay, again, another kind of work. So this locus is associated both with schizophrenia, right? When you have the deletion, it's associated with schizophrenia, and with autism. And oddly, if you have the duplication in this, it may be protective of schizophrenia, right? So there's some questions about, okay, this region seems to be interesting. The problem, though, is that there's still multiple genes in these CMVs. So we haven't gotten to a single gene yet. We've just gotten to an area of the genome where we can be fairly confident that the genes harbored in that region are influencing the trait we're interested in. So if that's the case, we have now, we have now thinking about rare sequence variation. So you not just a piece of meat, but a sequence of pieces of meat. So if we want to think about rare variation, I like to start by doing kind of the silly thought experiment. And the silly thought experiment says, okay, well, there are about 6.77 billion people on Earth, at least according to Google. And most estimates from whole genome sequence suggest that any individual has about 80 new mutations that haven't been seen before, right? So each of us has mutations that just are from us, right? So if that's the case, a little bit of multiplication will tell you that you have about 536 billion new mutations on Earth right now that didn't exist in the last generation. Okay. And if you divide that by the number of points of the genome, you get about 162 variants per each site on the human genome, right? That's pretty huge, except that this is actually wrong because some of the areas of the genome are inconsistent with life, right? So you can't have every variant at every point along the way. But it's not to tell you exactly this is the case, but it's to give you a sense of how much rare variation there is out there and what kinds of things we could think about with it. There's a growing realization that rare variation is actually pretty important. So this is a histogram of the first 1,000 people we did whole genome sequence on in my group. And what I've done is I've modeled the minor allele frequency versus the number of alleles that you have at that location, or at that frequency, right? And so what you're seeing is that 67% of the total variation in the genome has a minor allele frequency less than 0.01, right? That means that when you're doing GWAS studies, you're missing the vast majority of the variation that's out there. Okay, so so that leads us to the rare variant hypothesis. It's a substantial component of quantitative trait variation is due to rare variants, right? So that's the hypothesis, and that larger effect sizes could be identifiable, which is kind of what we showed before when we were looking at the effect size. But typically, the only way to find these variants is to do exome sequencing or whole genome sequencing. Okay. Whoops, sorry, wrong way. So do we have evidence for rare variation influencing diseases associated with brain? And so one set of evidence comes from schizophrenia, where you have this paper from Sean Purcell and colleagues, where they looked at exome sequencing in schizophrenia in about 2,500 individuals with schizophrenia and 2,500 controls, and they identified a bunch of rare variants in the patients that seemed to be associated with risk for schizophrenia. The problem is, if you are expecting a variant to appear only once in 10,000 people and you've only studied 2,500 of them, you're not going to get enough copies to make a statement about any one gene. In that case, what they did was aggregate the genes together and suggest that the genes that seem to have these rare deleterious variants were in voltage-gated calcium ion channels, the ARC scaffolding proteins, and postsynaptic signaling. Okay. Probably the best data we have is actually from the work in autism, where they did a design where they took parents of individuals with autism and then their children, and they did exome sequencing, and they searched for those things that were different in the children that were de novo mutations in the children relative to the adults. And by doing so, they were able to talk about a large proportion of the genetic variation associated with risk for autism, about 30% in a simplex collection, right? But that pointed to a likely distribution of about 400 genes and an additional set of Menson's mutations in a partially overlapping 400 genes. So you have a large search space, but you have a search space. And so we begin to make progress here. So what happens then in rare variation in kind of normal people? So you haven't selected them because they have an illness, but you want to know whether or not the rare variation affects genes or cognition. So here I'm going to show you one example from some of the work that we've been doing in Texas. This is part of the genetics of brain structure and function study that I do with my colleague John Blangero. It's an extension of the San Antonio family study. We have actually at this point about 1,950 individuals from big extended pedigrees that were selected for randomly, and we have whole genome sequence on them. So the questions are going to be, can we find genetic influences affecting cognition or brain, and how does that work? So the first thing we did was to take all of our cognitive measures and do what is, in essence, a genetically informed principal components analysis. So what we did is we took the genetic correlation between each cognitive measure and every other cognitive measure, and then we broke it down into sets of factors. And so we showed that you have a verbal memory factor and a working memory factor and a spatial memory factor, and they're all heritable. And based on some work that we were interested in, you can show that they're associated with the liability for schizophrenia. So we wanted to take this spatial memory factor, that one that had the highest association with liability for schizophrenia, and see if we could find a gene associated with it using whole genome sequence, right? So what we did is we went in and we searched the exomes, so the areas of the genome that we think are involved specifically in creating protein, and we wanted to search through this because a naive search of the whole genome space is just too large. We just don't have enough samples to be able to kind of define that. So we wanted to kind of reduce our search space. So we looked only in exomes, and additionally, we only wanted to look at those variants that are known to, or that have some prediction of being damaging, of influencing the protein that's present. Although I give you a list of different possible strategies you could use. So we searched all non-synonymous variants, which was about 25,000 in about 700 individuals, and we found a variant that was influencing the spatial working memory, or the spatial memory set. And particularly, I'm giving you the effects here. The important point is to note that the beta which is, in this case, is represented in standard deviation sizes, is about 0.7. So it's moving. If you have this variant, you've reduced down, your ability on this factor is reduced by about 0.7 of a standard deviation. Okay. What's interesting about this allele is that in our sample, about 2.8% of the people had it because we have big families, right? But in 1,000 genomes, the prevalence of this allele was less than 0.01, right? Okay. And we had 39 individuals who had it, right? That's why when you're trying to find rare things, it's useful to study families because families share a lot of their genetics, and it's more likely you're going to find rare variation throughout. This is particularly associated with a mensis mutation on PLEHG4. And all I wanted to show here is just that if you look at that particular variant, it's not simply associated with the tasks that made up the spatial memory measure. It's also associated with the working memory measures, the executive factors, as well as an IQ measure. So we have what looks like a gene that seems to be influencing cognition across the way, right? So what is this gene? Well, it's on 16q22.1, and it is a Purkinje cell atrophy-associated protein, right? It's gone by various names, and I will tell you that we actually then turned around and looked at it as in its association with anatomy, and it is influencing surface area. It is influencing cortical thickness, much less so. But unfortunately, FreeSurfer removes all your cerebellum. So I have no idea if it's influencing cerebellum yet. So I have to go back and figure out a new way of parsing my cerebellum so I can test whether or not this variant is influencing that or not. One last point that I'll make is that that location has a CMV associated with schizophrenia. It was published initially in this paper in PNAS in 2010, and that paper that came out just recently also showed an association with this. It just didn't quite make their statistical threshold. So, okay. So it's possible that we chose a cognitive trait because it was of its liability to schizophrenia. We searched the genome, and we've possibly come up with a gene that seems to be influencing it. There's still a lot of work to be done, and Jason will tell you all about that. But I'd just like to say these kinds of things are possible, and it's where I think the field will be moving in the years to come. I'd like to thank all of my collaborators, particularly John Belangero, who is slightly larger and slightly bolder than the rest of us. Thank you.
Do we have any rare variant? Would you mind microphone? Thank you.
Hi, this is a lovely overview of, I guess, a very complex field for the rest of us. A comment and a question. As a comment, if you think of autism, then what GWAS finds is probably more varying genes. And then if you look at the genetic correlation between autism and education, you have a positive correlation. And you could think of those people discovered this way as perhaps more or less functional. But then I imagine another spectrum of autism people have very severe effects and they're hardly functional. I was wondering if the rare effects are more genes are more affecting people in a very severe way, and you would perhaps get at the very severe phenotypic effects as well.
I don't think we have an answer to that in autism. There has certainly been an argument in the CMV literature let me restate that. I don't know of anybody who's made that argument in autism. I do know that in schizophrenia, that if you increase your CMV load, you have more deletions, you have a much higher likelihood of you have an increased risk of getting schizophrenia, right? So there is some idea like that, but I don't know if it works quite as easily as saying, it's a rare variant and that's going to be more associated with this more severe case. I don't think we have data like that at this time.
Okay, and I guess the other trouble with the rare variants is just finding them because they're so rare. And you were lucky in this sample that you had these families where they're clustered together. I was wondering if there's a feasible, say, if you really understand this one rare variant and the mechanism and possibly you could then try to find other genes who would have a similar mechanism and try to see whether they have an effect.
Right. So from my perspective, what you show when you do something like this with a rare variant is just simply that you've gotten a bigger effect size. So you're more confident about the fact that it's probably important. But from this statistical effect, now you need to go and do biology. Because if you don't do biology and you don't do biological manipulation, it's going to be very hard to convince Nature to publish your paper, right? And so since our real job here is to convince Nature to publish our paper, that I think is part of what we have to go forward with. And to teach us all how to get Nature to publish our paper, we have Jason Stein.
You can't.